All. This is Dr. Mubin Sayed from drbean.com. Welcome to one more show. So thank you very much for allowing me to go to FLCCC conference and take these days off. It was a wonderful time. I presented there as well for methylene blue mechanism of action and the mitochondrial health. I have done that discussion here on Dr. Bean, plus I've done that discussion for FLCCC, long story short, as well. So it was a great thing, and I took a lot of pictures. So I saw today that Dr. Paul Marik and Dr. Joe Varun will discuss or recap the FLCCC conference on this Wednesday in FLCCC Zoom. However, if you like, after this talk, I can come back for a chit chat and I can have you go through the pictures that I took while I was there. So just say yes or no in the comments, and that would tell me if we should do the chit chat or not. Secondly, the discussion that I want to do today is about the spike protein. And the spike protein, I did this discussion a few days ago of the study from Harvard Medical School and Boston Children's Hospital, where I believe it is Dr. Zoe Swank and her team that were showing full length spike protein in the blood of the children that had myocarditis. Same Zoe Swank has also shown spike protein in the blood of the patients who have long COVID after the COVID infection. That was a rather earlier discussion and study. This is more recent. So while I was at the FLCCC conference, and there were a lot of discussion about spike proteins, a lot of questions that I got, not at this stage, but when I was roaming around, a lot of questions that I got was, why is the spike there? After the infection or after the vaccine, why is it there? And what are the possibilities of the side effects of it? So I thought today I would just focus on the possibilities of the spike being in the blood after the infection or after the vaccine. So I think this is understandable that after acute infection or during the acute infection or soon after the vaccination, spike could be found in the blood. However, months after the infection or the vaccination, why is spike still present or found in the blood of some? And I also want to say this. I saw this message given multiple times during the FLCCC conference as well, that it is not seen, the spike is, and the side effects are not seen in everyone. That is why not everyone is vaccine injured or not everyone has long COVID from COVID. And so the speculation that we doctors were doing when we will sit down together and have, let's say, some food or snacks or just talk, we were making this speculation that at some point there will be genetic factors that will be revealed, that there are some people who had genetics which would actually allow them to not even have symptoms. So this is primarily Dr. Ryan Cole, he's a pathologist, so he was discussing this, and he used the term HLA. So HLA antigens are specialized antigens that we find in human tissues, and because of those presence or absence or type of HLAs, various tissue behave differently. That is how the blood typing and tissue typing occurs. So maybe we'll find various HLA types of various human beings where COVID doesn't say anything to them, that cannot even infect them successfully, or keeps itself asymptomatic, or the patient handles it so well that they stay asymptomatic, or become acutely COVID infected, or have serious outcomes, even death. Similarly, for vaccine as well, at some point we'll find out that there must be some folks who overwhelmingly respond bad to spike and some who don't. At the same time, Dr. Paul Marik made a comment which I think is really important as well. And he was talking about the contaminants in the vaccine batches as well. So maybe at some point we'll find out that there are vaccine batches that may have been contaminated. And those contaminants were causing damage in some people. And we were just saying, well, this is spike. So, and there may be a combination of all of those things. So with this some of the context, let's look at some studies and the presence of the spike. And the primary takeaway today, the thing that I want to explain today as much as I can, is various possibilities, the mechanisms that may be responsible for the spike protein to be present in the blood after a long time. We have already done this discussion of how spike protein causes damage and how much damage it can cause. So I want to focus on why is it present. So with this, let's look at, so Barbara says, good question, Andy. So Andy says, how do we get rid of the spike? So I have done many discussions, Andy, about that as well. So let's do this. Let me present the mechanism so this talk can stay clean to that side. Then as we do the chit chat for the FLCCC pictures, if you like, maybe after that we can do another chit chat of how do we get rid of the spike, or we just mix these two topics afterwards. So let me stay to the topic. So this is drbean.com. You will actually be happy that drbean.com now has ChatGPT integrated with it right on the main page as well. And you will be also happy, those who are becoming a part of Dr. Bean, we are spending $40,000 to develop diabetes lectures and plus cardiovascular management lectures. So those lectures would start appearing on the site in the next few weeks, and it would probably be a great thing. My plan for those lectures is to separately sell them for CME. However, whoever has access now, there is a link in the description that gives you access to everything. They will continue to have access to those newer lectures as well. Okay, so that is the little commercial for Dr. Bean. Here, let's start. This is a very good write-up. This is an article. People with long COVID may still have spike protein in their blood. So M. Gregory says, welcome back, doctor. Thank you very much. Good to be back. So this is in Scientific America, and it is actually interesting to read. In here, they have discussed with Zoe Swank, this author, Harvard Medical School, Boston, Massachusetts, USA, Department of Pathology, Brigham and Women's Hospital, Weiss Institute of Biological Inspired Engineering. So that is Zoe Swank. They talked with her and discussed that why is the spike protein after infection. So this very first study is the persistent circulating severe acute respiratory syndrome coronavirus spike is associated with post-acute coronavirus disease sequelae or long COVID. And this is their first study. This study that I discussed a few days ago, that the full-length free spike protein is present in the circulation of the children with myocarditis, that is also by the same team, or at least by the same authors, Zoe Swank, same lead. So they have been looking at spike for various cases. So this is the first one. And if you just want to have an overall idea of what they're saying in this is a good article. Then here, as I was just showing, this is the first study where they are showing that spike protein is present in the blood of people, infected people, even after months. So let me just show others as well. This is 23rd March 14, circulating spike protein detected in post-COVID mRNA vaccine myocarditis. This is what I presented last week. And here, what they saw was free, full-length spike protein present in the blood circulation or in the circulation of children who were suffering from myocarditis. There were other children as control who also were vaccinated but did not have myocarditis and also did not have spike protein. So this is also from the same team. Then this is a related editorial for the myocarditis and spike protein discussion, which I actually showed last time. I'm going to explain these three mechanisms. So here they're trying to explain that what could be the mechanisms for the spike after the vaccine to be present in the circulation months after the vaccination. So I'll go over this. And then immune imprinting, breadth of variant recognition, and germinal center response in human SARS-CoV-2 infection and vaccination. So this is another study where they are seeing, or they observed, spikes present and the antigens present in the circulation of people who were infected or people who were vaccinated months after the event. And they were talking about germinal centers and how this happens there. I have actually done this discussion in detail a few months ago. This is actually 2022 March 17 study. So I have actually discussed it a few months ago as well, or quite a few months ago. So having these in front of us, I want to first start with this study. This study, circulating spike protein detected in post-COVID-19 mRNA vaccine myocarditis. I have done the study. I'm going to go over these three mechanisms because what is interesting is that these three mechanisms were asked again and again. So let's start. First one, they said possible, these are hypothetical explanations of why spike may still be present in the blood months after the vaccine. And now this is not a overall hypothesis, or this is not a curiosity only that spike is present. This team has now shown deterministically that spike is present for the, at least in the children's blood who were suffering from myocarditis. So that is not a hypothesis anymore. Now the question is, why is it present? And here are three hypotheses. Number one, prolonged existence of mRNA that aids destruction. Now, this number one point actually can be explained by this study of 2022, March 17, where they saw that even after 2 months of the vaccination, the germinal centers, so if I can draw as well, and I don't have pre-drawn things, I came back last night from FLCCC and I just were lazy today. So what happens is, imagine this is a deltoid muscle. And in this muscle, the vaccine is given. Then what happens is that the lymphatic flow, lymph from this tissue, will bring the antigen being whatever is the offending agent, antigen to the local lymph nodes. So now in the case of deltoid muscle, the local lymph nodes are going to be lymph nodes in the axillary area, the lymph nodes in the supraclavicular areas, and on the side of the cervical areas. So these are the areas where the lymph node present there will be called local lymph nodes. So now in these local lymph nodes, when the antigen is brought, for in this case, spike protein or messenger RNAs or LNPs, when they're brought in here, then what will happen is that in the lymph nodes, there will be proliferation of the cells. Lymph nodes contain follicular dendritic cells. They contain incoming macrophages. They contain incoming fluids. They contain incoming broken antigens that are just flowing in the fluids and are passing by from here like a river passing with the contaminant and trash in it, like water bottles in it. Plus the follicles are the places where, lymph nodes are the places where T cells and B cells reside. They live there. So when an antigen arrives in the follicles, then the T cells and the B cells, they will pick up that antigen and start proliferating. That means they start increasing in number, and then they would start differentiating as well, which means they will become more specialized to handle this specific antigen. Sky Frog is here. Hello, Sky Frog. S says, we tried to warn you. Nobody listened. Okay, so let's continue to discuss our points here. So now that we have the lymph node, and that has gotten this antigen in it, the germinal centers are where the germination is occurring, or where the proliferation and differentiation or activity is occurring. So in these germinal centers is what they're talking about, that they found that the spike protein and messenger RNA was still present, even after months. So for example, in case of spike protein, this is 2022, March 17 2022. So we have discussed it like more than a year ago. In case of vaccine, if you see here, histological analysis of the draining lymph nodes, that is similar as local lymph nodes. Draining lymph nodes are where the fluids are draining from a specific tissue, shows marked impairment of germinal centers in severe COVID-19 compared with messenger RNA vaccines. So vaccines do not impair the germinal centers, while if there was an infection, then the local lymph nodes become markedly impaired. That means there is just destruction going on within the lymph nodes as well. Higher quantities and persistence of spike antigen accumulated in the germinal centers of mRNA vaccines and detectable vaccines RNAs in germinal center for up to 2 months after post second dose. So higher quantities and persistence of spike protein antigen, that is spike protein, accumulated in the germinal centers, that is the area of activity that is in the lymph node, of mRNA vaccines and detectable vaccine RNA in the germinal center. So spike protein is present and the RNA is present. That means the spike is being manufactured in the lymph node, not just in the deltoid muscle. So this is what they observed. And this is happening, let's say, 2 months after. So if now I go back to those 3 mechanisms for why the spike protein may still be present in the blood, the first mechanism, as you saw here, was the prolonged existence of mRNA that evades destruction. So of course, in this case, for 2 months, the mRNA was present, only then it was present for 2 months after, right? Where was that study? This one. Higher quantities and persistence of spike antigen accumulated in the germinal center of messenger RNA vaccines and detectable vaccine RNA in GCs for up to 2 months. So the first case to put at rest is that when the spike protein, when the messenger RNA is given, the messenger RNA is not only in the deltoid, it has gone to the local lymph node as well. And the local lymph node now is holding on to that messenger RNA even for up to 2 months and making spike proteins from that. So that is the mechanism number 1, especially for vaccine. If you see here, they did not say that they saw the same mechanism for infection. Why? If you see, histological analysis of draining lymph node shows marked impairment of germinal center in severe COVID. So in case of severe COVID, these germinal centers are actually impaired. They have become disabled. They are not functioning. I hesitate to call them infected, but they're impaired. That is the word they use here. So because they are impaired, the concept of messenger RNA sitting there or the virus sitting there or the spike proteins being formed there is more difficult. However, in the case of vaccine, they are clearly seeing higher quantities of the spike protein being built over there, plus the messenger RNA being present there. So mechanism number 1 done. And I would request to keep today's discussion to these mechanisms, because that was the largest number of questions I got for all the days I was there, that, hey, what is the mechanism? Why is the spike present? So number 1 done. Now let's look at mechanism number 2, increased dose delivery of messenger RNA. So the second mechanism, this was with the vaccine. They're saying that in a child, so let's say this is a child. Let me see if I can pull this drawing board a little closer so I can draw better. So let's say this is a child. So what's happening is that this is a child. So the dose that is given, so let's say this is the dose given, and this is the same dose, let's say, that is of an adult size 2. So this dose is so much increased for the child, so much more for the child. The child then continues to make spike proteins for a longer period of time because child is not able to clear that vaccine quickly and rapidly enough. And there are many possibilities for why not. For example, one possibility is that the children's enzyme that do hydrolysis. Hydrolysis mean to pour water on something and break it down. Just like when we want to soften something, we put water in it and then we kind of mush it and we soften it and break it. Similarly, our body does that as well. When it is trying to break something, it kind of adds water to it. It causes hydrolysis of that and breaks it. The enzymes that perform the function of hydrolysis and help with breakdown, child doesn't have sufficient quantity of hydrolyzing enzymes. That is maybe one possibility. Another possibility is that the child surface area compared to the child's tissue area sizes or volumes are lesser than an adult. So the same dose given for this lesser tissue amount or volume is actually generally higher dose. A third possibility is that the dilution occurs in the adult. So an adult has a larger overall volume of fluids compared to a child's total volume of fluids. So in that bigger volume of fluid of an adult, the amount of spike produced is diluted and is less compared to the same amount of spike produced if it is still the same, but because the fluid volumes are lesser, then relative amount or concentration would seem to be higher. So that is another possibility. Another possibility is that the adult's kidneys may be able to handle the spike's clearance fast enough compared to a child's kidney. And if you said, hey, where are you pulling these mechanisms from? These are all the mechanisms that are written here in this hypothesis. This is the editorial to go with that study that I mentioned before, this study by Zoe Swank, circulating spike protein detected in post-COVID-19 mRNA vaccine myocarditis. With this one, if you scroll down here, it says editorial, see page 877. This is that editorial. So the mechanisms that I am pulling and presenting are from that editorial or from the other studies as I just showed. So there is nothing here which I am hypothesizing. I am actually just showing what is there. Okay, then increased dose delivery, and I explained that how relative increase could be a possibility. And finally, possible role of anti-idiotypical antibodies. So anti-idiotypical antibodies or anti-idiotype antibodies, first I want to show you here, if I look for network theory of Neil Jenkins, local area networks. So Neil Jenkins immunology, potential neutralizing antibody discovered for novel. So if I then look here, so Jenkins, I do not know which one, but anyways, this is called the network theory by Neil Jenkins. And there is Dr. Bill Murphy, Dr. William Murphy, and anti-idiotype antibodies. Anti-idiotype antibodies here. So this is very important. If you're watching, you can actually look at this title here, a possible role of anti-idiotype antibodies in SARS-CoV-2 infection and vaccination by Dr. Bill Murphy and his team. And I have actually discussed this in detail as well. Actually, he was so impressed with my discussion that he says he sends out my video to anyone who discusses this paper with him. In addition to we met as well and we had lunch together, and it was just an amazing thing. He has appeared on my channel a few times as well. So after this paper, we did not know each other before. So a possible role of anti-idiotype antibodies. This I have been discussing for a long time. And I'm going to once again try to explain this one today too. So back here, the third possibility is the possible role of anti-idiotype antibodies resulting in the attenuation of neutralizing spikes. So let me first give you an example here, and then I'll explain this. Imagine there is a bad actor. There is a bad guy or bad gal. This is that bad actor, right? So there is a bad actor, and we have a protector. This is the, we should be angry guy. So we have a protector of our own that's going to fight with this bad actor and kill him. So this is our superhero. So maybe we should give it a little crown or something, right? So this is our superhero. Then what will happen is that when this bad actor is present in our body, we need to make these servicemen or women to go and attack these bad actors. Good. So that will be fine. You have a million bad actors and you make a million good actors or protectors, and these would go and attack these million, and these would clear out the situation. Think about it for a second. If we make another group of actors, we make them who go after these guys. Now what will happen is, let's call them, let's label them. Let's say the bad actor is A, the good guy to good guy to kill A is B, and then there is this weird guy C who is killing the good guy B. John says, give him a hammer. Which one should have a hammer, John? Right? So now we have three characters in this discussion. This A is the actual offending agent. B is the protector. C is the one that neutralizes the protector. Now the concept, John says, is that a water gun that the C guy has? No, he's just like doing punchy, posture. So what is the role of the C guy in here? The role is this. Imagine our body encounters these bad guys, bad actors, 10 million of them, right? And so these are the A's. So this is a group of A's. Body will do what? It will make group of B's, right? So it will make a bunch of group of B's. And so they are all getting ready, these B's, to go and fight with these A's. Now, body thinks that, hey man, I do not know how many A's or the offending agents or antigens I have. So I have really no clue how many B's or these antibodies that are going to attack them we should have. So body decides, you know what, I'm going to just make millions and millions of them. But then body sits down and says, oh man, if I made so many millions of them, I'll have a problem. I will have some of these B that may just be present here in huge quantities, and they will not be used up in breaking these A's, or they will not be used up in fight. And now these extra fighters that we made, the antibodies that we made, will attack my own tissues and start causing local tissue destructions, and it will become very difficult to handle these extra militia force that I have generated. So body says, you know what, for these extra ones, for protection purposes, let me make a lots and lots of C as well, whose job is to pick up these extra B's and neutralize them. So that means the B that our body is now making fiercely, antibodies, a group of B will become engaged in the fight with A. So good, that is all good. They would take care of A. Then the remaining group will become engaged with C. And then that is also good because now the extra B's will be engaged with C and we are all good. Some of this is here, other group is here, nothing is free, nothing is running around, and nothing is killing each other, meaning tissues. So body is very smart and it is doing this. This is called the Neil-Jenkins theory. However, the hypothesis is that when this antigen comes in, let's say the virus or the vaccine, body starts making B and starts making C, and B, instead of fighting with A, B become entangled with C. The love story starts here. And the A is just standing there saying, ta-da, I have no one to kill me because these guys are just fighting with each other. They are complexing with each other. And the end result is the neutralization of A is not happening. Why? Or the neutralization has become weaker. Why? Because the neutralizing antibodies are now stuck with anti-idiotypical antibodies. This is not what body wanted, but this is where body ended up. That is what is the third mechanism, the possible role of anti-idiotype antibodies resulting in the attenuation of neutralizing spike antibodies. Attenuation meaning they are just binding here with the neutralizing ones, and so neutralizing capability is reduced for the offending agent. Now, I'm going to explain this in a non-cartoony way to explain what these anti-idiotypical antibodies are. So here, I think you would like the cartoony way anyways, but so imagine that we have spike protein. We know that spike protein has an S1 part and an S2 part, correct? So, and then spike protein has a receptor binding domain, correct? So let's say this is the receptor binding domain. That is where it binds with the ACE2. On that domain, there is a specialized area that is called receptor binding motif. I have never drawn it this way, but today I'm going to draw it this way. So this is these black rectangular shapes, three of them, are receptor binding motif, RBM. Who has heard this from me so many times that it is kind of on your tips? So here we have this receptor binding motif. This motif is actually small. It is 7 to 13, 14 amino acids long. So it's not a very long piece of spike protein, but this is the part of the spike protein that binds with the ACE2. Actually, that this receptor binding motif does that. Now, when we make an antibody against this one, what we'll do is, so this will be the neutralizing antibody because that antibody is going to bind here. So of course the antibody's binding part has to be a complement or the opposite of this binding shape. So I'm going to make a red one over here. That would be binding shape like this. So if I make the other side free, it would be one rectangle goes here, then the other rectangle goes here, then the other rectangle goes here. So this is the antibody. So if you see, this is really important to see that this variable region of the antibody, why do we call it variable region? Because antibodies have a similar structure. However, this part of them is different, and that can bind with various kind of pathogens or viruses and fungi and stuff and various parts of them. This is why we call them variable regions. So far so good. We have antigen in black. We have the receptor binding motifs neutralizing antibody, which is in red, and it is a complement of the shape of the receptor binding motif. Good. Now, think about it with me for a second. I'm going to make it in green now, actually black. those C guys, these guys, this guy. So body in its abundance of caution says, you know what, I'm also going to make another set of antibodies that can neutralize this antibody. Now imagine you are the one, you are the body who's going to make this another set of antibody. I have put that request to you to say, hey, you are a manufacturer of antibody, can you make me an antibody that can destroy this antibody? And you'll say, okay, I'll make you an antibody that would bind here. And I'll say, no, 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 it should not bind here. This is, by the way, it is called the fraction constant or FC part. So you say, well, I'm going to make an antibody that will bind with the FC part of your antibodies. And I say, no, no, no. All of my antibodies have FC parts. If you made an antibody that binds to the FC part, it's going to attack every single antibody. I don't want you to attack every single body. I just want you to attack this specific antibody. You say, okay, I got it. I'm not going to make an antibody against FC. I'll make an antibody against the light chains. So light chain is this part. I say, once again, time out. No. This light chain is also common in all antibodies. Why will you make an antibody that attacks the light chain? It will attack then everything. And you say, okay, so we need to have a very unique part of this antibody that I can make antibody against. And we say, yeah, sure. This variable region is unique. This region is not present on every antibody. Every antibody has this variable region which is different on them. It's okay, very good. So I'm going to make you an antibody that is going to be able to bind with this variable region. So now we both are happy. Why? Because you're going to make an antibody for me that will bind with this variable region of this specific antibody. And when we will present this new antibody in our body, it would clear out any free antibodies which were supposed to work with the or block or neutralize the spike protein. These antibodies, the black ones that I drew, are called anti-idiotype antibodies. Why are they called anti-idiotype antibodies? Because they are attacking the idiotype of the other antibody, or the variable region is called idiotype. So they're attacking the variable region of the other antibody. Good. So let me ask you a question. Look at this part of the spike protein and look at this part of this anti-idiotype antibody. Do they look similar? Do they look similar to each other? Paul Wolf says anti-antibodies. That is correct. So generally we call them anti-idiotype antibodies, but anti-antibody is fine as well. So if I go back here, for example, they'll say role of anti-idiotype antibodies. Similarly, if we go to Dr. Bill Murphy's, he would also talk about anti-idiotype antibodies. So that is the term we use. So this guy, the black guy over here, these are the black antibodies are I wrote it here, anti-idiotype antibodies. Do you see a similarity of anti-idiotype antibodies binding region and the receptor binding motif of the spike protein? They are the same. Because on this antibody, they both can bind. Right? M. Gregory saying, I want to learn Qigong and tapping. M. Gregory, what's up? We are doing medical lecture. So Paul Wolf says, motif is mirror image like DNA, RNA. Correct. So this guy and this guy are very similar. Because of that, now if I said to you for a second, I said, you know what? This one can be present in large amounts, and not only that, free. then that is a problem. Even more scary thing is this. Now, if I do not make their binding region, just say that when the antigen comes in, so let me make a bunch of blues, blue, blue, blue. These are all spike proteins, vaccine or COVID. Our body readily says, all right, I'm going to make all these reds, which are the antibodies, neutralizing antibodies to attack the blues. But instead of they being able to attack the blues, because our body made a lot of these anti-idiotype antibodies as well, they rushed in the anti-idiotype antibodies and bound with the red. What will happen to these guys? The spike proteins, will they be attacked? No. Why? Because their attackers, the blue, are mostly busy with the black, which we produced. We produced the enemy of our own protector. So there are going to be very few reds that can go and attach with these blues. So the overall neutralizing capacity is now reduced. Neutralizing antibodies are actually produced, but they are neutralized by our own other anti-idiotype antibodies and a number of free antigens or spike proteins are now running around in the blood because there is nobody available to block them. Why not? They are actually present, but they are blocked by our own other antibodies. And ta-da, we have free spike protein. So these are three possible mechanisms of the spike being present in the blood circulation. I'm going to add a fourth mechanism. The fourth mechanism is actually also by Dr. Zoe Swank. It's not my proposal. She said that it is also possible in case of infection, the virus itself is hiding somewhere. And she thinks that the virus may be hiding in the central nervous system or it may be hiding in other tissues. And because it is hiding there, it is causing the damage, but that is kind of subclinical type of damage. And all we know is that these spike proteins and the other viral antigens are running around in the blood. These are kind of markers of virus hiding somewhere. And so it is a persistent virus itself. And before anyone attacks me and says, Mubin said this thing, which is wrong. Here, she is talking about, so if you see here, Zoe Swank, persistent circulating severe acute respiratory syndrome, coronavirus 2 spike is associated with post-acute coronavirus disease. And if you see here, she would discuss so we measure the concentration of SARS-CoV-2 antigen present in full length. And so let's say reservoir here. They say if viral reservoirs persist in the body of past patients, long COVID patients, why do we not also detect N protein in more patients? And she gives an answer for that. We found that the spike could be detected even if the neutralization levels were high. So furthermore, PASC is a heterogeneous syndrome, possibly dependent on the tissue in which the viral reservoir persists. So this is active viral reservoirs could cause PASC symptoms, but circulating spike may also give rise to symptoms. And we know that spike itself can bind with the ACE2 and cause these issues as well. So in this study of hers or theirs, they talk about viral reservoir being another possibility, which could be the one to give rise to continuous presence of the SARS-CoV-2. Now, viral reservoir for sure is known to be present in patients with immunosuppression or immunocompromise. So South Africa, patient of HIV with persistent spike after 246 days. I actually presented this study. I hope I can, this is 2018, so this is something else. Um, if you, I should have said, South African patient of HIV persistence COVID SARS-CoV-2 spike. So I think it is somewhere over here. I have actually done this. That was a female who had HIV but not controlled, and she had the presence of SARS-CoV-2. She became acutely ill for 2 weeks. The symptoms resolved, and then 246 days after, she still had active SARS-CoV-2 present in her. So we know that in certain patients, SARS-CoV-2 for sure can be present. However, in this study, Dr. Zoe Swank is postulating that this may not even be that somebody needs to have the immunosuppression or to have immunocompromised state. It can be a possibility that the virus is uh, living in them. So now we have four mechanisms in front of us. Either the virus is present or there is another which is for S1 part, and that is Dr. Bruce Patterson's study that we also have been discussing for a long time, where he says that the S1 is hanging out in the monocyte for even up to 15 months after the acute event, and that event could be infection or the vaccine. That's a different, he says he does not find the full length, but he finds S1. Dr. Zoe Swank says that we did not find a lot of S1, we found a lot of full length spike proteins. And they postulated that maybe it may have been easy for our body to take care of S1 or N proteins more easily compared to the full length spike protein. Dr. Bruce Patterson actually postulated that maybe it is difficult for us to clear S1 from monocytes compared to the whole S2 parts. So there are all kinds of studies, but they're all trying to address the same one common observation, that spike protein is observed in the blood circulation for vaccinated or long COVID patients. Okay, so 47 minutes, I cannot believe it. I took your 47 minutes. But because this was the most common question, the presenters were discussing that how spike protein could be present and the side effects or the issues with the spike protein, but the basic underlying mechanism that what could happen. So it is possible that 2 months later there is a newer mechanism than these, but so far these are the mechanisms that are known or that are postulated. Correct. So Eric says PASC equals long COVID. Yes. So they call it post-acute SARS-CoV-2 sequelae. So that is the post-acute coronavirus disease. That's PASC. We call it long COVID for a long time. So it is long COVID as well. We have done this discussion many times that the long COVID itself is either the damage to the tissues. For example, somebody becomes severely ill and their lungs are damaged. And now afterwards, the lungs become scarred and they become shrunken and now they are oxygen dependent. So that is a sequelae of the lung damage because of COVID. But we are talking about mostly when we say long COVID, this is the COVID where the patient actually heals, becomes okay, and then a couple of months later, then they start having symptoms again, and that may be related mostly to immune system. So S says that Dr. Ryan Cole is the only one with a grasp on this and being honest about it. It is actually fun. Dr. Ryan Cole and I were traveling together back from the FLCCC conference. We went for a dinner together, and he was so complimentary of my work, and he said, every single day I watch your videos, so I know what I'm seeing in pathology, but then I need to understand the mechanisms behind that as well, and I watch your videos for the mechanisms. So that is so nice of him. So this is the discussion. So Broken says that there is a study about CBD. Yes, I saw the study that CBD can actually help bind uh, with the spike as well. So I'll do that. Broken, yes. Okay, so with this, um, I'm gonna hang up now. It's 50 minutes. I hope people can watch it. So please like, subscribe, and share. And there are links in the description to support this work if you would like to support this work. And there are links in the description to buy Dr. Bean Access before the access becomes different, as we are adding more lectures and creating courses. So that is the talk. I request you once more to please tell me, just say yes for the chit chat or not. If you would like for a chit chat, tell me and I'll come back and I'll show you. I'll take 5-10 minutes to compile the pictures I want to show you. I'll walk you through the FLCCC pictures if you would like, and we can do other chit chats as part of that as well. But please like, subscribe, and share. And chit chat, yes and no. I'm going to look at the answers. So bye for now.